Okay, thank you very much. I am Irene de Prado, and I will present the Silent project on bioaccumulation of poly and perfluoroalkyl substances, PFAS, in aquatic macrophytes from the Amazon River. PFAS are persistent organic pollutants composed by a hydrophilic head and a 4 to 16 carbon hydrophobic chain, which could be either totally or partially saturated with carbon fluoride bonds, forming per or polyfluoroalkyl substances, respectively, which classifies them by families. They could also be classified by chain length, as long-chain PFAS are more bioaccumulated than short-chain ones. The carbon fluoride bond is one of the strongest in organic chemistry, making these molecules extremely stable, so they are called forever chemicals. They are ubiquitous and especially risky for aquatic environments due to their moderate polarity and high acidity constant. They are highly toxic, producing carcinogenicity, neurotoxicity, developmental toxicity, and many other harmful effects on organisms. Their commercialization started in the 1940s as Teflon, and nowadays they are used in several grease-proof and waterproof industrial and consumer products. PFAS end up in aquatic environments through urban runoff. The Stockholm Convention on POPs regulates the use of the most common PFAS since 2009. However, Brazil has a license for production and use of N-ethyl perfluorooctanosulfonamide, or ET-FOSA, which composes the pesticide sulfuramide. This can be degraded to PFAS via biological and abiotic mechanisms. Sulfuramide is mainly used to control leaf cutting ant populations occurring in livestock grazing pastures and agricultural fields. Nevertheless, both sulfuramide and urban sources of PFAS should be considered as important contributors to PFAS pollution in the Amazon River, its estuary, and the Atlantic Ocean, as it could be seen in this marine PFAS pollution map, where it increases next to Brazil. This research is inside a bigger project called Silent Amazon, which is the first study on the presence and distribution of anthropogenic contaminants through the Amazon River. The main objective of this study is to assess the occurrence and internal distribution of 27 congener PFAS in 3 aquatic macrophyte species, Eichhornia crassipes, Eichhornia zurea, and Poteria rotundifolia from the Brazilian Amazon River. Moreover, the partial objectives of the study are to analyze and compare the total PFAS families and chain length concentrations per internal distribution in the macrophytes between the 3 macrophyte species, and per spatial distribution gradient from inland towards the coast and at different habitat types, urban versus open river. And finally, to compare the results of this study with results of previous studies in macrophytes from other countries. This research is relevant because this is the first study on bioaccumulation of PFAS in the Amazon River. Macrophytes add a fundamental structural complexity to aquatic ecosystems, increasing biodiversity and stability of communities, and providing juvenile fish with food and shelter. They could be used also as bioindicators of PFAS pollution in aquatic ecosystems and as proxies of chemical discharge into the marine environment. 17 macrophytes of the 3 species were sampled through the Amazon River at different sampling sites during November and December 2019. And for each individual, subsamples of leaves, stems, and roots were divided, if available, and stored at -20 degrees centigrade. Furthermore, for the analytical method optimization and validation, macrophytes of similar species as the one in this photo were taken in Spain. The following analytical method was used for PFAS extraction from the Amazon samples. Plant samples were freeze-dried, ground, and spiked with a solution containing PFAS isotopically labeled with carbon-13, which were used as surrogates for the analytical procedure. Then 2 extraction cycles with acetonitrile diluted in water were applied to the samples, and after that, wax cartridges were used for solid phase extraction. An extra cleanup with envicar carbon powder was applied to the elutions and samples were filtered before analysis by liquid chromatography tandem mass spectrometry. Data acquisition and integration of chromatographic peaks was done with Agile and MassHunter quantitative data analysis software, and furthermore, 3 procedural blanks without matrix and without internal native internal standards were analyzed with this protocol to evaluate possible contamination during PFAS extraction process, and when detected, their PFAS concentrations were subtracted from the ones in Amazon macrophytes. 3 different extractants were compared in a previous experiment, and acetonitrile diluted in water was the most suitable one. Furthermore, a validation experiment defined the limit of detection, limits of quantification, and matrix effects by spiking samples at different with PFAS at different concentrations of PFAS at different times during the experiment, and with and without matrix. Further data analysis was performed with Excel and SPSS, and maps of the sampling sites and distribution of the PFAS concentrations in macrophytes by QGIS and ArcGIS. Several results were obtained from this study. Ponderia rotundifolia bioaccumulated significantly more PFAS than the other 2 species. Previous studies demonstrated a higher PFAS bioaccumulation potential of Ponderia genus Ponderia, in contrast to genus Eichhornia. As it could be seen in the graph of the left, Ponderia rotundifolia bioaccumulated significantly more PFAS in aerial parts of the plant or stems and leaves, especially perfluorocarboxylic and carboxylate acids, or PFCAs, as it could be seen here in yellow. It is remarkable that perfluorooctanoic acid, or PFOA, one of the most industrially used PFAS, pertains to this family, but it was not the most detected compound, as it was recently regulated by the Stockholm Convention. Genus Ponderia could increase the size of its root system in more eutrophic ecosystems like the Amazon River, in contrast to genus Eichhornia. Moreover, a previous study from the Silent Amazon project, which analyzed water samples from the Amazon River at the same sampling site as the plants where the plants of this study were sampled, revealed that PFCAs were the most abundant PFAS in water. So a bigger root system could have made Ponderia rotundifolia absorb more PFCAs. The majority of PFCAs detected in this study are short chain, which means that they are more easily translocated to aerial parts because they are more hydrophilic than long chain PFAS. No significant differences were found in concentration of individual PFAS between plants from open river versus the ones living in urban areas. PFPA was the most abundant PFAS in Amazon macrophytes, followed by, as you can see here in yellow, PFPA is perfluoropentanoic acid, and followed by perfluoroundecanoic acid, or PFUNDA, which is shown here in light green. This shows that industries are switching to production of shorter chain PFAS as long chain PFAS are more strictly regulated. However, short chain PFAS can also be dangerous for wildlife. A significantly higher total PFAS bioaccumulation was found in plants from urban areas in contrast to the ones living in open river, which was expected due to the numerous anthropogenic sources of PFAS in towns and cities. For the 2 habitat types, a significantly lower bioaccumulation was found in roots in contrast to aerial parts. In addition, bioaccumulation in roots was significantly higher in open river macrophytes than in urban ones. This could be explained due to a higher concentration of PFUNDA, a long-chain PFAS, occurring in open river macrophytes in contrast to urban ones, as shown in the previous slide. Related in roots because they are more hydrophobic than short-chain PFAS. Finally, bioaccumulation per different PFAS families and chain lengths was not significantly different between the 2 habitat types, suggesting that a same source of PFAS occurs in two habitats, or the two habitats have the same source of PFAS. No significant correlation was found between total PFAS concentration per macrophyte sample and its distance to the Atlantic coast. If we look at this bioaccumulation distribution map, a significantly higher bioaccumulation was found in plants from urban areas, for example, Manaus and Manacapuru, in comparison with open river habitats. Macrophytes from less populated cities around the middle of the transect, for example, Santarém, had lower PFAS concentrations than the macrophytes from more populated cities, for example, inland cities, for example, Manaus, and the ones closer to the Atlantic coast, for example, Macapá or Belém. Sorry. Bioaccumulation of PFAS in Amazon macrophytes was lower than in similar aquatic macrophytes from other countries. The ones in China and Sweden presented the highest PFAS concentrations at 3 orders of magnitude higher than the ones measured in this study. This could indicate that the use of sulfuramide in Brazil is probably not the main source of PFAS pollution in the Amazon River. And as I said before, a switch to production and use of more short-chain PFAS could be seen in Brazil. In conclusion, short-chain PFCAs are the most abundant, or were the most abundant PFAS in aerial parts of Pontevedra rotundifolia. Plants from urban areas were more contaminated by PFAS than plants from open river habitats. Hence, the studied species could be a good proxy for mapping PFAS pollution through freshwater habitats and also its discharge into marine and coastal environments. And these results also could guide PFAS pollution management directives in order to implement water treatment systems, especially in urban areas. PFPA was the most abundant PFAS in Amazon macrophytes, so a possible point source of PFPA should be investigated in future research in the Amazon River, in order to also assess its possible risks for aquatic ecosystems and human populations which rely on them. Finally, the use of sulfuramide in Brazil is probably not the main cause of PFAS contamination in the Amazon River. So other PFAS sources should be considered in order to implement these pollution management directives, for example, urban runoff, as I said previously. Furthermore, chemicals directly related with sulfuramide, such as ET fossa or N-ET fossa, are currently being analyzed in samples from the Silent Amazon Forest, the results of this study. Thank you very much for your attention, and I hope you enjoyed my presentation.